Any investigation of venom must start with the animals that have killed more humans than any other. Snakes. The scientist in charge is Bryan Grieg, the world expert on the evolution of snake venom.
It's a magnificent snake.
Collecting venom is laborious and dangerous.
All right.
For Fry, a day at the office can involve milking up to 50 different species of snakes. Every one of them a killer.
This is the Australian mulga snake. So this is a brown snake. What we've got here this time is a death adder. They're actually related to the cobras. So what we're doing is we're milking all of these snakes so that we can have the venom to study in the laboratory. You have to get the venom, obviously, from only one source. It's got a good little yield on him. That's something you see a lot of variation in snake venom, is the color of it.
Sometimes the process goes wrong.
Like I had one bite from one of these kinds of snakes where it bit, swung the fang, stuck it in, swung the other fang, stuck it in, swing, and just walked across my hand. I had a series of puncture wounds going across it. That was fun.
A dose of anti-venom saved Fry's life. But every year, 125,000 people die from snake bites. No anti-venom works for all snake bites because venom is so incredibly varied.
Venom is the ultimate in the movable feast, where not only do you get big variations from one species to another, but even within the geographical range of a single species, you can get quite pronounced differences in the venom composition, where they might have a different menu, you might say.
Fry has dedicated his career to working out why venom is so diverse and so deadly. Could its evolution be the answer? Fry already knew venom was a combination of deadly chemicals that worked in unison.
Think of it more of like basically a gang beating you to death with different shaped iron bars. It's a concerted, multi-dimensional attack on your body.
His quest to understand venom would take him 5 years. For Fry, just knowing it was complex wasn't enough. He wanted to know where all those chemicals came from and how they evolved.
It's a lot like sort of doing a molecular detective story. What we do is we break it down. We separate all the different components out into individual components, do a wide variety of tests and other analyses to find out what makes it work, what makes it kill, and how can we use it.
Fry first tested the effects of hundreds of different snake venoms, observing their terrifying impact on animal tissues. Some dissolved flesh like acid. Some made organs like the heart burst from the inside out. Others caused instant paralysis. He then used the latest technology, called liquid chromatography, to separate out the chemicals within each venom. On these graphs, each peak indicates a different killer toxin within just one venom.
So what we've done is we've separated this complex mixture into each of their individual components, where these have been collected into their own vial. We now have a fingerprint of this venom. So the snake venom proteins don't come out of thin air. What they are is they're mutated forms of natural body proteins, often ones involved in very important physiological processes.
So how does this mechanism work in practice with the most venomous snake of all, the taipan? To see the effects of its venom in action, Fry experiments on his own blood.
We've just taken a not small amount of my blood out, and we're going to use that. We're going to add taipan venom to one vial and leave the other vial alone, and then we'll be able to see the difference. So what I'm doing is adding the taipan venom to the blood. This is my blood. Without any venom, this is my blood on taipan venom. You can see what it's done to it. It's turned it into a solid clot. Now imagine this is my blood, but my blood actually inside of me. Obviously, this is not going to do good things. By making all those millions of tiny little blood clots, all your blood clotting factors are being used up. What this means is that you now can't clot, so you'll be bleeding out of your gums, your ears, your nose, everywhere else. It's not a nice way to go.
The taipan's venom evolved from proteins that formed clots in its own blood to heal wounds. Once injected, it reduces its victim's blood to a mixture of massive clots and a liquid that leaks from every orifice. When the taipan bites a human, venom surges into the body and peppers the bloodstream with millions of tiny blood clots. This triggers a cascade of deadly effects, shutting down the liver, kidneys, and lungs. All it takes is a fraction of a teaspoon. One mystery remains about the taipan. Out of the thousands of snake species on Earth, why is the taipan's venom so powerful? Fry thinks there's a reason for that.
So this is the Australian taipan, all of its legendary glory. Unlike any other Australian snake, they specialize on feeding on very dangerous prey items. Snakes normally don't try to go after these kind of prey, such as bandicoots and the native plague rat, because these are animals that could severely injure or even kill the snake.
In this footage, a plague rat can be seen taking a bite out of the taipan. That's why, over millions of years, the taipan had to evolve ever stronger venom in order to knock out its prey as fast as possible. Biologist Bryan Grieg had discovered how snakes had turned their bodies into bioweapons, engineering venoms of staggering diversity. And that diversity keeps evolving as snakes tailor their venom to suit their wide-ranging habitats, including even underwater.
The sea snakes are incredibly venomous snakes. And the reason for this is that in the aquatic environment, they don't have the luxury of tracking their prey. If the prey gets away, that's it. Someone else, like this little ray here, is going to make an easy meal out of it. So they need their incredibly toxic venom in order to make sure that they get their meal.
Sea snakes evolved from land snakes only 30 million years ago, and they're still refining their venom. They prove that snake venom is still adapting. But when did its evolution first begin? When Fry first set out to solve this mystery, it was assumed that venom was a recent innovation. It was all to do with snakes' extraordinary fangs. Fangs are hollow hypodermic needles through which venom flows under extreme pressure. This is how the fangs of a spitting cobra can propel its venom as far as 8 feet. They're so perfectly engineered to deliver venom that it was assumed fangs and venom had to have evolved together. But when Fry inspected the fangs of modern snakes, he found something intriguing. 3 major groups of snakes had evolved very different fangs.
You've got different lineages that have hollow front fangs, but of very different types, where the vipers, like the American rattlesnakes, swing on hinges and are very long. The cobras are short and fixed, while the stiletto snakes, they pop out sideways.
For Fry, this meant venom must have existed before the different types of fangs. Sure enough, when he and other snake experts across the world traced the evolution of venom back in time, they discovered the first snakes had no fangs, but they did have venom. Rather, they'd inherited their venom from an earlier ancestor. Fry decided to study them.
But they can be quite tenacious once they latch on. Now, better this than my hand.
Fry's theory went against common wisdom. Out of 5,000 lizard species, only 2 were known to be venomous, and they were considered evolutionary freaks. Fry decided to search for other venomous lizards. He zeroed in on the most ancient lizard species in the world, monitor lizards, such as the Komodo dragon. Komodos are fast ambush predators that can grow to the length of a car and weigh as much as a linebacker. The bites from their serrated teeth result in rampant infection that eventually kills their prey. Scientists had long thought this was caused by bacteria, 80 species of which are known to exist in the Komodo's saliva. But this explanation didn't satisfy Fry.
It never sat right to me about all this stuff about the toxic bacteria and Komodo dragons because, well, bacteria don't work that way. They don't cause rapid bleeding, muscle pain, and neurotoxicity within the first few hours unless it's some sort of escaped bioweapon or the Andromeda strain.
Fry wondered if there might be something other than bacteria triggering toxic effects in Komodo's prey. To find out, he would have to take a closer look. Using a brain scanner, he imaged the head of a Komodo dragon. It was then he found something that had been totally overlooked.
With the Komodo dragon, you can see a very large bulge running the length of the lower jaw. And with our magnetic resonance imaging, which is the same technology that you use to scan a brain to look for cancer, you can see the bulge there. Now, if you look into the beauty of technology, you look within it, you can see a very complex gland arrangement. And that's actually a venom gland.
Fry had discovered a venom gland, similar to that of snakes. Might there be other venomous lizards? There was only one way for him to find out.
So to decide what was happening, we ended up going out and collecting as many of these things as we could.
Fry's team swabbed the mouths of hundreds of lizard species for saliva samples. He isolated their active molecules and tested them on animal tissues. The results were remarkable. The molecules broke down the animal's heart muscles and toxified their lungs, the same way as the venom of certain snakes. This wasn't bacteria. This was a slower-acting form of venom. Fry's team eventually found snake venom components in more than 1,500 lizard species. He had rewritten the evolutionary record.
We started looking at what was actually the chemical composition of the venoms and found that there was several components that were shared between the lizards and the snakes. And this was the final proof that they shared a common venomous ancestor.
Venom needs complex, sophisticated biological engineering to inject it.
In order to have a venom, you need to have a way of delivering it as well. And there's been a lot of different ways that this has been evolved, whether it be the fangs of the spider, the barbs of a stingray, the spines of a stonefish, the spurs of a platypus, etc., etc. Each one has their own unique way of delivering it.
The chemicals extracted from all sorts of venomous animals are being converted into drugs. The same toxin of the taipan snake that disrupts the blood of its victims is being developed as a drug to stop excessive bleeding during surgery. Already, people with certain brain cancers are treated with radioactive versions of scorpion venom.
This incredible variety of venom is actually what makes it so useful. The absolute ingenious evilness that's gone into the design of it means that we can take these things, find out what makes them work, and then modify it to make it used as a drug, where basically nature has already done all the work for us. We just need to figure out what they've done.